Hello students, this is the second session of second unit, that is general characteristics of microorganisms. In the previous session or in the previous class, we discussed about the introduction of microorganisms, that is the definition of microorganisms, sources of microorganisms, and different types of microorganisms, how the microorganism is classified like bacteria, protozoa, algae and fungi and the virus, and also we discussed about the uses of microorganisms where all the microorganisms are used. Now we'll discuss about the general characteristics of microorganisms. Bacteria are the type of biological cell. They constitute large domain of prokaryotic microorganisms. The major morphological characteristics of bacteria mainly include size, shape, arrangement of the cell and structure. Okay, now we'll just see about the size of the bacteria. The bacterial cells, they are about 1/10 the size of prokaryotic cells and are typically 2 to 10 micrometers in length, and they are ranging from 0.2 to 1.5 micrometer in diameter. When we see about the shapes of bacteria, there are three main basic shapes of bacteria. They are coccus, that is round shape, they found in spiral shape, and they found in the bacillus shape, that is the rod shapes. Okay. If we talk about the arrangement of the cells, bacterial cells are arranged uh, in the different forms individually basis on the shapes of the cells. If we consider arrangement of the cocci, they are arranged in Diplococci, Streptococci, Tetrads, Sarcinae, Staphylococci. So, what is this Diplococci? Diplococci, they usually cocci divide in one plane and remain attached in pairs. So that is a Diplococci. We can give an example of Diplonemoniae. Okay. Streptococci. Streptococci, here cocci divide in one plane and remain attached to form a chains of different lengths. Tetracocci, here cocci divide in two planes at right angles to one another to form group of four. About Staphylococci, here the cocci divide in several planes, resulting in the formation of irregular bunches of a grape-like clusters of cells, for example, Staphylococci aureus. So like this, the bacterial cells are arranged. Here we can see the arrangement of the cells. This is a Diplococci, Staphylococci, Streptococcus. Okay. Similarly, the bacillus are also arranged. There are bacilli bacteria. These are either present singly or may be arranged differently. They divide in only one plane, and the cell formed may adhere to form pairs of cells or chains based on they are grouped into Diplobacilli. Diplobacilli, here we can see here. Here we can see the Diplobacilli. Bacilli cells are arranged in pairs. Streptobacilli, here the bacilli cells are arranged in form of a chain. Example, Streptobacilli we can give. And the palisade like that is these are the cells arranged side by side like matchsticks, an angle to another. That is example for palisade like bacilli, it is the Corynebacterium diphtheria. And trichomes, when the cells of the chain have much larger area of contact with each other, it results into the formation of trichomes. And about spiral shaped bacteria, The spiral-shaped bacteria occur into two forms, that is the Vibrio and the spiral. Vibrio and the spirillum. Vibrio, these bacteria usually show a single curve and they are shaped. For example, Vibrio cholerae. The spiral-shaped bacteria, these are longer, relatively rigid, curved forms. Example for this is Spirillum minus. And if we see the structure of bacteria, they lack organelles such as chloroplast and mitochondria. They do not have the true nucleus found in eukaryotic cells. Instead of their DNA double strand that is continuous and circular is located in a nucleus, means they don't have a double strand DNA. They have the continuous and circular like DNA. The nucleus is an irregularly shaped region that does not have a nuclear membrane. Here we can see the structure of bacteria. See, here we can see the capsule. Capsule is called as the outer layer. The green color what we are seeing here, that is a capsule, that is called as capsule. It is gelatinous layer present on the surface of some bacterial cell wall. Chemically, these are made up of polysaccharides, and in some bacteria it is made up of polypeptides of one or two amino acids. This capsule, it is common in pathogenic bacteria. It protects the cell against temporary drying and destruction by phagocytic action of a host cell, and also it protects cell against the action of antibacterial agents. And next to this we can see the cell wall here. The green color you can see here now, this is the cell wall of the bacteria. Usually bacterial cell has thick rigid cell wall. It prevents the cell from expanding and bursting. It is often made up of peptidoglycan, and we can see the flagella here. Flagella, these are a fine whip or hair-like structure. These are unbranched, wavy helical appendages that produce through the cell wall. The protoplast, here we can see the inside, the bluish color cytoplasm or the protoplast. The contents of bacterial cell lying within the cell wall together from the protoplast, and this protoplast, it consists of outer plasma membrane. This is the cytoplasmic membrane, or we can say the plasma membrane, cytoplasma, and this is cytoplasma, and the nucleoid. This is the irregular shaped nucleoid we can see here, and pili-like structures. Here outside this capsules we find some hair-like structures. These are called as pili. These hair-like appendages on the outside of the cell allow it to stick to surface and transfer the genetic material to the other cells. What these pili will do, they will help the bacterial cells to stick on the surfaces, and also they help to transfer the genetic materials. Okay, and this can contribute to the spread of illness in the human also. So this is about the structure of the bacterial cell. Next is, that is all about the morphological characteristics. The next characteristic is the cultural characteristics of bacteria. Usually bacterial growth in and on food is extensive enough to make the food unattractive in appearance. If the bacteria is growing on the food, means definitely the food will look very unattractive. The pigmented bacteria, they cause discoloration on the surface of the foods. We can find the discoloration like pink color, we find black color, and also green color, some white color, like that discoloration of the surface of the foods we can see. And film may cover the surface of the liquids. Film like a layer like cover may also develop on the surface of the liquids, and growth may surfaces slimy, means if the growth of the bacteria is there, that surface will be slimy in nature. Okay, the growth throughout the liquids may result in undesirable cloudiness or sediment. And if we see the physiological characteristics of bacteria, the growth and activity of bacteria in food, it includes chemical changes. The chemical changes which include the hydrolysis of complex carbohydrates to simple ones, hydrolysis of proteins to polypeptides, amino acids and ammonia or amines, hydrolysis of fats to glycerol and fatty acids, oxidation reaction which are utilized by the bacteria to obtain energy from the foods. They yield the product as organic acid, alcohols, aldehydes, ketones, and some gases. Okay, and if we see the reproduction of the bacteria, usually the reproduction of bacteria is takes place by the binary fission. Binary fission, it is an asexual form of reproduction in which cell continues to grow until new cell wall grows through the center forming two cells, and these separates making into two cells with same genetic material. That is by the binary fission. The another kind of reproduction in the bacteria, that is transfer of genetic material. Here cells, they acquire new genetic material through the processes known as conjugation, transformation or transduction. What happens here is these processes can make bacteria stronger and more able to resist the threats such as antibiotic medications. Even by the spores also the reproduction takes place when some types of bacteria are low on resources. They can form spores. The spores hold the organisms of DNA material and they contain the enzymes needed for germination. Thus, the reproduction of bacteria will takes place. And there are many uses of bacteria. We find bacteria are used for many purposes, like many of the bacteria in the body, they play a very important role in the human survival. Bacteria, it helps in the digestion. You can say the digestive system breaks down the nutrients such as complex sugars into forms and body can use. The bacteria take in nitrogen and releases it for plant. They use when they die. Plant need nitrogen in the soil to live, but they cannot do this themselves. Bacteria can break down the organic compounds, and this is very much useful for the activities such as waste processing and cleaning up oil spills and the toxic wastes. Along with the uses, we also find some hazards of the bacteria. Means there are different useful bacteria and hazardous bacteria, or we can say the harmful bacteria. What hazards we find here is some types of bacteria, they can cause diseases in humans such as cholera, diphtheria, dysentery, uh, plague, pneumonia, tuberculosis, typhoid, and many more. If the human body is exposed to bacteria that the body does not recognize as helpful, the immune system will attack them, and this reaction can lead to the symptoms of swelling and inflammation that we see, for example, in an infected wound. So with this, we can summarize this, that bacteria are type of a biological cell. They constitute a large domain of prokaryotic microorganisms, and the major morphological characteristics of bacteria includes size, shape, structure and arrangement of the cells. Bacteria, they reproduce sexually. so that the two daughter cells that results from binary fission and have the same DNA as a parental cell, they also reproduce sexually by transferring the genetic material. The bacterial growth throughout the liquids may result in undesirable cloudiness or sediment, and these are there are many uses of bacteria, uh, and also we find many hazards of the bacteria, and these are the references which I have used for this section. Thank you.